Hello, everyone, and welcome to this package demo on SpatialExperiment. Before we start, just a few housekeeping, which you've probably heard repeatedly already, but bears repeating. If you have questions for the speakers, you can put them in the Q&A tab, and we will be moderating questions at the end. You have the ability to upvote questions, so you can go into the Q&A and vote for questions that you want the speakers to answer. As you can see, there are a variety of emoticons that you can use to let the speakers know your thoughts. And if for some reason you have to leave early, the video for this session will be available a couple hours after we're finished. So without further ado, I'm going to hand it over to Dario Righelli, Lukas Weber, and Helena Crowell, who will be talking about spatial experiments.
Thank you. So welcome, everyone. I'm going to share the screen. Okay. So welcome. I'm Dario Righelli. I'm a postdoc at the University of Padova in the Davide Ristos lab. And today, with Helena Crowell and Lukas Weber, we are going to talk about the spatial experiment infrastructure for spatial transcriptomics data. This is the page, the bookdown page for this workshop. Here you have some available resources. The website, of course, the Docker image that you can run on your own laptop with this instruction, and some links to the packages that we are going to show you today. So, and also the articles, because this is a three-part workshop. I'm going to introduce some concept of spatial transcriptomics. So we define with spatial transcriptomics technologies all those technologies that are able to join detect transcript stratification with their spatial coordinates, referring to an understudied tissue. We distinguish at the moment two main families, the molecular-based and the spot-based technologies. The first one is focused on the molecules, so the transcripts. And the main idea is that they are able to detect the transcripts in multiple temporal snapshots at cellular and subcellular level. We identify in this family, for example, the SecFish and the MERFish and some other technologies. And the differences inside this family are mainly due to the way they detect the transcript coordinates and the number of detected transcripts. For example, in the SecFish and later the SecFish Plus, they use temporal barcodes for the temporal snapshots that I mentioned before, and with multiple hybridization rounds for the molecules, combining image microscopy and fish approach. So in this first figure, we have to imagine this first figure as a snapshot, as a one singular temporal snapshot of our experiment. These are our multiple hybridization rounds, where the transcripts are detected thanks to luminescence, to a color. So at the end of the hybridization rounds, so in this case there are n hybridization rounds, we can identify each transcripts with its own their own barcode color identification. So there is no amplification for the transcript in this case. Indeed, the transcript abundance for these kind of technologies is due to the number of times that that each transcript is detected inside that specific temporal barcode. So, for example, in this case, the mRNA1 is detected 3 times thanks to the barcode associated to it, and the mRNA 3 two times, and the mRNA 4 and 2 just one time. And this can be applied directly on a tissue slice, thanks to the image microscopy. So you can zoom and detect the portion of the slice to detect the transcripts. The number of transcripts changes depending on the technology. For example, for the SecFish at the beginning, the number of transcripts was not so high, but with the SecFish Plus, this technology is able to detect thousands of transcripts at each round. So this technology can be comparable to a whole transcriptome technology. On the other hand, the spot-based technologies have barcoded reference spots. They use barcoded reference spots. So the coordinates are associated to the spots. So the main difference from the previous one is that the coordinates were associated to the transcripts, while in this case they are associated to the spots. The spots changes depending on the technology. In this case, we just mentioned the 10x Visium and the slide-seq. And so inside the family, it changes the type of the spots, the size also, and that's what I remember at the moment. For example, in the 10x Visium, they use these chip where there are all these holes inside. The holes are the spots. So we already know the number of the spots inside on the chip, and we place a tissue slice on the chip, and so we automatically know which are the spots covered by the tissue. And while we take the snapshot with a microscopy of the tissue placed on the chip, at the same time inside the spots, a cDNA amplification starts, and so inside each spot we have a whole transcriptome amplification for each spot. Also, we have, as mentioned, an image reference in this case, and some other details will come up later in the parts of the workshop taken by Helena and Lukas. So for handling all these kind of families of technologies, we developed the SpatialExperiment class, extending the single-cell experiment class. So as usual, we have the observation on the columns and the features on the rows. And we extended this class with these data structures: the spatial data, which stores the spatial metadata for the observations; the spatial coords, which stores the spatial coordinates in matrix form; the image data, which is a data frame for storing the image metadata, and also the images, so we can directly work with images for the technologies such as the 10x Visium. And finally, we extended the assays with a dedicated assay, the molecules assay, which is designed for the molecules-based data, and it's based on the bumpy matrix class. This bumpy matrix is a sort of multi-dimensional data frame, and maybe we will take a look at it in the other parts. So again, this is the instruction for the Docker image if you want to run it on your laptop. And finally, just one last mention, one extra resource: if you want a multimodal SecFish dataset and you want to use it for your own aims, we developed this. We contributed to the single-cell multimodal PyContactor package, providing this SecFish and single-cell RNA-seq dataset. And here is a link where you can find additional details, and these are the instructions for install the package and also to to take a look at the VNet. So if you have some questions, otherwise I will leave the stage to Helena for the hands-on on the class.
Okay. I think, yeah, you need to stop sharing. Okay, is it working?
Yep.
Okay, so I'll just keep this open here as a reference for myself and also everyone else. And we thought to easily demonstrate the class, we'll just use some Visium data as it has a very predefined structure. So typically when this comes out of the Cell Ranger pipeline from 10x Genomics, you get exactly these outputs with fixed file names, and that's our starting point. So I'll just copy this one chunk and the rest I can hopefully live code. So I'm just loading the package and specifying the path to where we've made available some data inside of the package. That's really all that chunk does. Why is it so slow? Okay, so here we just have these two paths where our data is stored. And let's just look at one of these. So we have a subdirectory called spatial. That's this one, and a subdirectory of the raw feature barcode matrix. And then I'll just look into these. Path. In this first directory, we have our barcodes. So that's our observation metadata, our features, and the matrix, which are our counts for each of the spots. And in the spatial directory, we have a file that has the scale factors. We'll see later what those are, a low resolution image, and then the spatial coordinates and array positions, etc. Now, because this is quite complex data, I think, it would take quite some lines of code to read this in. So we made a function for that, specifically for reading 10x Visium data. And what we specify here is the paths to where our files are located, what we want to call these. That's the section 1 and section 2 up here. We say that we want the low resolution image only because we can imagine there can be many different images. We want to load the image, so we say load equals true. And then we want to read in the raw features, so that's type equal raw. Alternatively, this could be filtered, which is also returned by the pipeline, but here we're reading in the raw data. Okay, and because our data is not in HDF5 format, I'm sorry, this should be sparse, and then the data should be raw. And this should be called sample typo. Sample IDs. Okay, finally I got it. Okay, so now we have our spatial experiment, and just as a spot check, this inherits from the single cell experiment class. And of course, the single cell experiment itself inherits from the summarized experiment. So this part up here is nothing new. We have our assay data, which is the counts. So every row here is a feature, every column is an observation. In a single cell experiment, those are cells. In our case, they're barcoded spots. We have some metadata associated with every feature and some metadata associated with every cell spot. So that's nothing new. Now, what is new about the spatial experiment, as Dario already mentioned, are these 3 slots. So first of all, we have some spatial data that comes from the Cell Ranger pipeline, which first of all says if a spot has been mapped to the tissue or not. And this is based on this image that has some staining underlying it. So this is something that comes out of the pipeline. And for every spot, we get the position on the chip where this spot is located. so the array row and column. Secondly, we have spatial coordinates. So these are the pixel in full resolution. So these are all integers. And what we can, I can just maybe plot this really quickly. So that's kind of what it looks like. This is now coming from 2 different samples, but still, this is kind of what our spots currently look like. Of course, this is the downsample data set. In reality, the whole chip is has spots all over it. Okay, and maybe what's worth mentioning is that these slots here sit in an internal slot of the object. So they're only accessible, they should only be accessed with the accesses we provide. And we've implemented so that they have a hierarchical structure. So on the lowest level, we have our spatial coordinates. On the next level, we have the spatial data. But here we can maybe say, okay, we also want to include the spatial coordinates. And then on the highest level, we have the call data. And here we can say, okay, we want to include everything. And that will give us really all of the metadata associated with every spot, including the sample ID, the spatial metadata, and the spatial positions. And this is maybe useful if we want to plot something using ggplot. We really want to access all of this data at once. Alternatively, we could access everything and then bind it together into a single data frame. Okay, and maybe the most interesting bit is then this last spot here, which is the image data. And as Dario mentioned, this is a data frame, capital D data frame, where every row is an image entry that has a sample ID associated with it, an image ID, and a scale factor. And this scale factor is important if we wanted to overlay these spatial positions with the image, since these are corresponding to the full resolution image, which is huge, and we're not storing that, we would have to multiply them by the scale factor to map the spots to the image that we're plotting. And then in here, in the data column, sits the actual image. And those are spatial images, which is a class that we implemented. And maybe just a quick word on that. So the spatial image class itself is virtual. It doesn't have anything, but it gives birth to 3 separate classes: the loaded spatial experiment, spatial image, which we have here. So the image is actually loaded into R. We can have a remote spatial image. So this can be sitting on a URL in the internet or something. And a stored spatial image, which sits somewhere in the computer, and we have the path to it, but we haven't yet actively loaded into R. And this is useful if we want to store real images somewhere, maybe online, but when we work with the object or we want to export an RDS object, we don't necessarily have to put these images into it and increase our file size unnecessarily. So I'm just going to extract one of these images here. So as I said, this is a loaded spatial image, 600 pixels times 476. And what we can do with that is convert it to, for example, a raster object. And that's just a matrix where every pixel now has an associated color with it. And that's pretty cool because we can just plot that, and then we get our image. Alternatively, we can also access this using the image raster accessor, which also goes directly into the spatial experiment and grabs this raster object for us. So that's exactly the same thing. And then also there's another accessor, the image source, which would tell us where the file is located or the URL. But since we loaded this image, there is no source here in this case. Okay, now say we were interested in adding an image to our object. So I'm just going to add an image here from Google. So this is just a internet site with a JPEG on it. And we can say add image to our single-cell experiment. The image summarized experiment. The image source is our URL. We want to add it to section 1, and we call it something. Okay, I knew I was going to get that. Then we also need to specify a scale factor because this is required, and I'm just going to put NA here. Okay, now we have a new image entry here. So the image is now stored inside our object, and we can again plot that. And this time I have to specify which image I want to plot. I want the one that I just added. And we get a picture of a very cute dog. Okay, then because this is not very useful, let's remove that again. So we can do remove image, and again specify the image we want to remove, and the image is now gone from up here. So yeah, these are just some functions that took longer to develop than they look like when I demonstrate them, sadly. Okay, finally, just a word on maybe the sample ID. So this is something we chose to fix, meaning it's required to be in the object. And the reason for that is that if we have multiple Visium experiments, we get identical positions for all the spots. So everything is really mapped onto each other. And it's really crucial to have a sample ID to know which spot comes from what experiment. So if we try to do sample ID null, this doesn't do anything. We still keep our sample IDs. Similarly, if we set the call data to null, we will remove everything but the sample IDs. They will always stay fixed. If we put something weird into it, like we just call everything X, we'll get an error that says the number of unique sample IDs is 2, but you only provided 1, so this doesn't work. And similarly, if we just shuffle our samples, we get that this doesn't work because there's no unique mapping to what we put in. So how can we do that? Let's just grab our sample IDs, and now I'll just paste a letter to them, something like this. So now our sample IDs are something like that. And then indeed, this works. So now we've done a valid replacement with a uniquely mappable sample ID mapping. And now we've just relabeled our samples. Of course, we can do a better relabeling than this. Okay, and since I have 2 more minutes, I wanted to maybe just quickly jump to the molecule-based way. So this is Visium data. And I'm just grabbing, I'm sorry for this, but I'm grabbing an example from the documentation. I'll run through this in a bit. So what we're doing here is we're sampling 1,000 molecules for 50 genes and 20 cells. And for each molecule, we sample what gene, what cell, and what's the X and the Y position. So this data frame here is 1,000 rows. Every row corresponds to a molecule. And what we can then do with Aaron's bumpy matrix package is to convert these to a so-called bumpy matrix. We'll see in a bit what that is. And we say, okay, we want these X and Y positions. Our rows are the genes and our columns are the cells. And then we get this bumpy data frame matrix. It has 50 rows and 20 columns because we have 50 features and 20 cells. And what we can see here is the first entry is a data frame. So there's 2 molecules in the first, for the first cell and the first gene, there's 2 molecules, and these are their positions. And then for the second molecule and the 10th cell, there's just 1 molecule. So we can go on like that for the, let's do something like that. Oh, I'm not getting a different output. Okay, they're all 1. But you get the idea. So this is a 3-dimensional data frame, but every entry can have a different dimension. So we can have 10 molecules in one and 5 molecules in another. And that's what the bumpy comes from. And from that, I'll just create a spatial experiment. Missing one thing. Now, how do we get the counts from that? That's pretty simple. We can just tabulate the genes and the cells. And then I convert that to a matrix. Okay, so I've just counted how many times do we get every gene and how many times do we get every cell. And these would then be our counts. So similar to what we had before in the Visium. And we can then create a spatial experiment from that, where we say our assays are the counts and our molecules is the bumpy matrix. So this is here, we have 2 assays now, and we have an accessor for that, which is molecules. And there we get our molecules. So now in a single spatial experiment, we're storing both the aggregated molecule level data, which are just counts, where every row is a gene and every column is a cell, but we're also storing the molecule level data inside of the object, which is pretty interesting if you think about it. And with that, I'm handing over to my colleague, Lukas Weber.
Okay, thanks, Helena and Dario. So I'll just share my screen. Okay, can you see that on the ST example data package page? Okay, so yeah, so thanks for the really nice overview of the structure and the technology. So in this, so this is going back to the web page here in the articles part at the top. This is now chapter 3, ST example data package. So in this part, we're showing a package we made called ST example data that has some pre-formatted data sets from various technologies in the spatial experiment format, and then showing how to load those and to make some plots. And we've done this for use in examples, tutorials, demonstrations, and then for our own examples as well. And also there's a second package, 10x Visium data, prepared by Helena, which has several more data sets from the Visium platform. So in this ST example data package, in this version, there are 3 objects, the Visium human DLPFC, Visium mouse coronal, and Siegfish mouse embryo. The Visium DLPFC is the one that I'll use in this example. This is a single sample of human brain dorsolateral prefrontal cortex measured with the Visium platform from a paper that we've published. The full data set from 12 samples is also available through Bioconductor from the spatial LIBD Bioconductor package developed by Leonardo Cayeta-Torres. And here we're just using 1 sample from that for the examples. So this ST example data package can be installed from Bioconductor as well in the devel version with the current version of the objects, or you can also get it from GitHub if you're working in release. And in fact, there's an additional argument there needed to get the latest version of the objects. On the GitHub README page, I've included that if you're installing it. So yeah, get the installation instructions from the GitHub README. Then now I'll switch over to RStudio to show how to load the objects. Yeah, so this is the same page that I was showing before, but in the RStudio R Markdown version. So these objects are stored in Experiment Hub, so you can load Experiment Hub and query it for all the data sets stored from the STExample data package. If you're these are the release versions here, which have the _313. If you've got the devel version, which has the latest ones, it doesn't have those underscores and has new Experiment Hub IDs. Then, loading the packages, which I've loaded previously here as well. For the latest development version, we have this helper function, load data, and otherwise, if you're using the devel version, you can use the Experiment Hub accessor functions. There's more information on these different loading methods in the vignette, and especially showing how to access the Experiment Hub IDs directly as well. And then, as Helena showed just before, we can look at the object. So we've called it SPE as previously, has all the different pieces. The assay, the dimensions here, so rows and columns. And then assay names for this one, there's a single assay called counts. And the row data has the information about the genes, so gene IDs, gene names, feature type is also in there as a standard column. And then the col data, this has the general column data not related to the spatial information. In this data set, we have a column called cell count, where we've counted the number of cells per spot in this Visium data set. Then also ground truth layer annotation for cortical layers in the human brain from the paper that we've published there, and then the sample ID. Then the second part is the spatial data. These are the additional spatial metadata information pieces, like the in tissue, whether each spot is over tissue or not. And finally, the spatial coordinates themselves, which is a matrix here, X and Y coordinates, and the barcode IDs as the row names. And the image data as well, as Helena just showed. Now we can do some plots of this data set. We've prepared another package called GD-Spotbased, which is currently in GitHub, and we'll put that on Bioconductor. I think we'll submit it next week. So yeah, it can be installed from GitHub right now as well. Then loading that. And one of the functions here is called plot spots. This one we can use to just do a very quick check that the orientation of the image is as we expect. And from previously having looked at the images here, that is what we expect. Then in this data set, those columns in the column data that I mentioned before, we can use to annotate the spots. For example, using the one that's called ground truth. We can put our ground truth cortical layers on there. Zoom in, then the spots are a bit more nicely formatted. And this is just showing the spots over tissue with the tissue equals 1. Then secondly, we've also got a more flexible function in there that's more specific for the Visium platform only. That one's called plot Visium. So this will let us show the histology image background as well. So plot Visium, just like that, shows us both the histology image as the background and then the spots overlaid on top. So that's very nice. And then again, we can annotate this in various ways, and there are a lot more options there as well. So here, we're using that same ground truth column that we had before, and highlighting also the in tissue spots. So then that's what this looks like. Okay. Then so that one does have quite a few additional options in there as well. I won't go through all of those. And that's as far as I'll go with the quick intro demo. Then we left an exercise here as well if you want to try out some of these objects and plotting functions. One idea would be to try generating a plot in XY coordinates as I had previously, but showing something different for the annotation, such as the total counts per spot. So then think about how to calculate those total counts from the different pieces in the object, the row data, column data, and assay table, and then where to store that. And then back to the plotting function, how to actually provide that to the plotting function as annotations. So have a look at the help file for whichever plotting function you end up using. And then another exercise is also generate a plot in XY coordinates with annotation showing the expression of a specific gene that you're interested in. And in this data set, some of the ones that are very interesting are SNAP25, MOBP, PCP4. So have a look at the object, where those genes are stored, where the names of those genes are stored, how to find them, like in row names, column names, and so on, or row data, col data. Then get the expression values of those of that gene specifically, the one that you've chosen, and provide that to one of the plotting functions. And if you're looking for the solution later, it's in the source files of this workshop, so you can find it there. So I'll go back to the workshop. So all of this material is here on this page as well. And the other two chapters that Helena and Dario showed are here from the drop-down menu at the top. And to install this, so I think all the instructions should be there at the top. And there's also the Docker image, as Dario mentioned, which has a current working version of everything together. So I'll stop sharing, I think. And now we have some time for questions.
Yes, thank you very much, all of you. That was very, very interesting. We do have a few questions that they were answered in the Q&A, but I'm just going to show them briefly on stage in case people were not kind of monitoring the Q&A because they were so enthralled with what you were showing on the screen. So Rohit asked this question: is it specific for 10x or can it be used for nanostring data too? And Dario responded. I don't know if you want to expand on that, Dario, or if that is...
I don't know the nanostring data, but in general, if you have any kind of data with coordinates associated to it, so for the features or for the observations, you can use this class to handle this kind of data, and then to apply also methodologies in bioconductor developed for the spatial experiment or for the single cell experiment, as someone else was asking in the. I don't have anything to ask. If Helena or Lukas have something else.
I agree. So we haven't tried nano. Sorry, Dario, your microphone is quite loud. So we haven't tried nanostring, but yeah, as long as the coordinates are there, I think it should be possible. And yeah, maybe we can try this.
Okay, and this one came in during Helena's presentation, and Edward responded with this. Do you guys want to expand on that or on the Space Ranger count?
No, that's true. That there's a full resolution image that can be, I don't know, 0.5 gigabyte, or it can be really big. But in the example packages that we provided, we always just use the low resolution image, which is sufficient to generate like a figure that would fit on a PDF, I guess, and to display in R. So we're never using the full resolution or the high resolution images at the moment. But that could be, of course, done.
Perfect. All right, this question has not been answered yet. So anybody who would like to take it on?
So that's a great question, and basically yes. So that's a key thing that we've tried to ensure that, by inheriting from single cell experiment. It means that the entire suite of single cell methods out there can be applied to spatial experiment objects. I think that's that's a really crucial feature of spatial experiment, that it lives within the bioconductor ecosystem in this way.
Okay, thank you. And another one from Edward.
this is also related, I think, to the other one. I'm not sure what plot spots returns, but in these plot Visium one, there's currently no input argument to do this, but we could easily add it. So currently the spots are just sized just so that they don't touch each other and you would see the image behind. We could easily have an argument to make that adjustable. It could also be manipulated once the plot is created because it is a GG plot. So yeah, that can be done. Or you could change the alpha of the points to make them transparent or similar.
Okay, thank you. And one more.
Yes, exactly. Yeah, they return ggplot objects, so then you can do you can add any other ggplot layers after that. Yeah.
Yeah, and maybe just a comment that plotting the image, the histology, and the data on top of each other is not so trivial, which is why we have these plotting functions to do it. It's quite.
And in fact, in plot spots specifically, I think we expose the size argument for the geom point as a specific argument. Yeah.
Okay, thank you. Are there any other questions?
I might just add to my previous answer on the single cell experiment compatibility. So there, yeah, if you're on SCADA or something, then it would store the outputs in cold data. So then you can continue from there. Yeah.
Okay, we are just about at time. So if there aren't any other questions, I'm going to thank our speakers for this wonderful presentation. The next session starts in 15 minutes, so you can take a break. You can head over to the lounge and chat with people. I hope you're enjoying the conference so far today, and we will see you all soon.
Thank you.
Bye.